Hi, I'm Toto Olivera from the University of Utah, and I'm going to tell you about cone snails. So in a previous lecture, I talked about how cone snails hunt fish, but today I'm going to tell you a little more about the various strategies that cone snails use to capture their prey. So the venoms of cone snails have peptides that are very powerful toxins that target specific ion channels. And what we have been discussing are the 100 or so species of cone snails that hunt fish. As it turns out, these fall into several classes. And the species that we've encircled are cone snail species that use a very specific strategy. And that's illustrated by this video. And so there's a cone snail that's buried under the substrate, and it has sniffed a fish. And now it's extending its proboscis towards the fish. This is a real-time video, so you'll see how efficiently this strategy can work. And so the cone snail is extending its proboscis, and it's going to strike and tether the fish. And as you can see, this all takes place very, very rapidly. And the fish is engulfed and digested. And so the cone snail has a meal that's probably good for a week or so. So this is a typical strategy of a fish hunting cone snail. Now, it turns out that there are 700 species of cone snails. And the venom of each species has different components. And so this was unexpected. You would think that in most gene families there's a lot of conservation, but in the genes that encode the peptides expressed in the venom, what we find is that at the species level, there's essentially a completely different set of peptides. And so that means that this is really a very large pharmacopoeia of biologically active peptides. So there are lots of peptides in one venom because they use their venom to capture their prey, to defend against predators, and to deter competitors. But the question is, why are there different peptides in each species? And the reason is because each species presumably has its own ecological niche. And therefore, in interacting with prey, predators, and competitors, in each ecological niche, there's presumably a different spectrum of prey, different predators, different competitors. And therefore, when a particular species becomes fit for that niche, it has to therefore change the spectrum of peptides that are present in the venom. And so it is those genes that are expressed in the venom that make a particular species maximally fit for a specific ecological niche. And that's why these genes really begin to evolve very, very quickly at the species level. And we understand how that evolution occurs. So we know enough about how the peptides are made so we can explain exactly how that evolution occurs. So the peptide is first synthesized as a precursor, which is larger, and it's called a pre-propeptide, and it has three regions: a signal sequence, a propeptide region shown in green, and the mature peptide region. And to generate the mature peptide, a proteolytic event cuts off the boundary between propeptide and the mature peptide. And so this shows you a particular example of the sequence of a precursor. And the peptide itself is about 25 amino acids in length. And as you can see, the precursor is about 75 amino acids. Each peptide in the venom belongs to a gene superfamily. And the gene superfamilies are interesting in that each gene superfamily has two characteristics that identify it. First, a particular signal sequence. And within a gene superfamily, this signal sequence is incredibly conserved. And then in the mature toxin region, all the members of a particular gene superfamily have a characteristic arrangement of cysteine residues. And so in the example shown, there are two gene superfamilies. And in the S superfamily, you see 10 cysteine residues that are always separated from each other. And in the A superfamily, in most peptides, there are only 4 cysteine residues with 2 of the cysteines right next to each other, as shown in the examples. And if you look, you can see that the signal sequences within the S superfamily are conserved, and in the A superfamily, they're also conserved. But if you compare S to A, there's no similarity at all. So if we hone in on the S superfamily, here are 2 peptides, and you can see the signal sequences are absolutely conserved. And in fact, remarkably, there isn't even a silent mutation between these two sequences at the nucleotide level. While if you look at the toxin region, which is shown here, you can see that all the cysteine residues are conserved, but everything in between has mutated. There's no sequence identity at the amino acid level. And so what's going on here is that this arrangement of cysteine residues is important for the structure of these peptides. And so each superfamily essentially has a particular structural motif, which in most cases can be conferred by the cross-links between the cysteine residues, the disulfide bonds. So within a particular superfamily, this shows you the O superfamily, the cysteines are arranged as shown in the diagram, where the first cysteine is always disulfide bonded to the 4th, the 2nd to the 5th, etc. And so that cross-linking gives all the peptides in this superfamily more or less the same shape. However, hypermutation occurs in the mature toxin region in the hatched areas. And so what's essentially the net result is a story that's very similar to antibodies. You have conserved structure, and you have very rapidly evolving function or binding specificity because of these conserved and hypervariable regions. And so the conserved regions lead to the conservation in structure. The hypervariable regions lead to the rapid evolution of new function. And so by having these gene superfamilies that are organized in this way in the venom, what happens is the venom can very rapidly evolve new binding specificities, new physiological function. There is a problem. If you have small peptides and they're disulfide bonded, let's say if you have 6, then you have 15 possible ways to arrange the disulfide bonds. Only one of these is the correct biologically active form. And what's remarkable is the snails in their venom have essentially the correct isomer every time. We don't yet understand how they do this because some of these peptides are as small as 15 amino acids. And if you try to fold them in vitro, you always get a mixture. And so this shows you 1 correct isomer and 5 examples of wrongly folded forms. And in vitro, if you try this to fold them, you always get a mixture. But the snails always get it right. And so our speculation is the following, that each part of this precursor has a different function. And of course, it's the mature toxin, which is the biologically active gene product, and that's what gets injected into the envenomated animal. However, we speculate that the reason the signal sequence might be so highly conserved is because it may dock the precursor to a particular locus on the endoplasmic reticulum and package it with chaperones that ensure that the folding of the peptide, the correct disulfide bonding, is maintained. Otherwise, it will not be secreted. In the case of the propeptide region, that seems to be the docking sites for enzymes and factors, including post-translational modification enzymes. So Here's a diagram of the most common gene superfamilies, and each superfamily then has diverged into families that target different ion channels. And as I said, a feature of the mature peptides is that they have post-translational modifications. And there's some pretty unusual post-translational modifications in these peptides, including the carboxylation of glutamate, a flip post-translationally from an L to a D amino acid, which is amazing that this occurs in about 5% of the peptides. Bromination of tryptophan, sulfation of tyrosine, etc. And the way this seems to happen is that the enzyme that modifies a particular amino acid has a docking site on the propeptide region. So then the enzyme binds, and when it binds to that docking region, it then modifies a specific amino acid. In the example shown in the cartoon on top, tryptophan is modified to bromotryptophan, and then a different docking site presumably is seen by an enzyme that recognizes a glutamate and adds a carboxyl group so that now you get gamma carboxyglutamate. And so this is how these modifications seem to occur. And that seems to be one function of the propeptide region. So that just gives you a summary of our insights from molecular genetics as to how these peptides are made. Now, the other thing we can use molecular genetics for is to understand how these different species are related to each other. And this shows you a diagram of about 5% of all cone snails and how those species are related to each other. And what you can see is that this tree has branches, and most of the branches are labeled with W, which means those are worm-hunting clades. Those labeled with M in green, those hunt other mollusks, so those are snail-hunting clades. But for the example that I'm going to discuss, there are 4 different groups labeled with F, and those, in fact, are the fish hunters. And it turns out that all of the species that I've told you about have been this branch, the F2 clade, and those are the ones that use the strategy that I just showed you. So what do these branches mean? What does it mean that you have 4 different groups of fish-hunting cone snails? Well, what it means in real biology is shown in the next couple of slides. Here's a member of that second clade. In fact, this is Conus geographus. It's a fish hunter. When you drop a fish in, it doesn't stick its proboscis out. It opens its mouth. And you can see that it has a humongous mouth, really quite an impressive mouth, as shown in the video. Here's another species in that same group, Conus tulipa, the tulip cone. And this snail has frills at the end of its mouth. And as you can see, it's engulfing a fish. So again, it doesn't stick its proboscis out. It opens its mouth, engulfs the fish. And only when the fish is in its mouth does it harpoon the fish and inject its venom. So this behavior is entirely different in this clade from what we showed earlier with the other fish-hunting cone snails. And so what's going on here? What's going on is that there are 2 general strategies for catching fish that the snails have discovered. You can use a harpoon and line, which is used by many, many fish-hunting cone snails, or you can use a net, which is used by the 2 species I've shown. And so if you're going to use a net and you have a lightning strike cabal, when you hit the fish, and now the fish is twirling at the end of the proboscis with very stiff fins, that's not a good idea if, in fact, the fish is already in your mouth. And so, needless to say, this type of snail does not have a lightning strike cabal. Instead, what these snails do is they apparently primarily hunt schools of small fish that are hiding in reef crevices at night. And so the snail approaches the school. It releases a little bit of its venom. And when it releases its venom, the fish begin to behave as if they're derived sensory deprivation. They seem less motile. They act very much like they're in an opium den. And so we call this group of peptides that induce this syndrome the nirvana cabal. Okay? And so these peptides that are released first change the behavior of the school of the fish, makes it easier to engulf the whole school. And so now the snail can capture the whole school, begin to harpoon them one by one at its leisure, and they do not have a lightning strike cabal. So this is a different strategy altogether, requiring a different set of toxins to be evolved. And so the nirvana cabal puts the potential prey in a sedated and quiescent state. So you might ask what are members of the nirvana cabal, and we've identified a few of them. And so all of these quiet down neuronal circuitry, make it less active. And so now the question is, can we use these, apply them, to quiet down overactive neuronal circuits under pathological conditions? And so what are examples of overactive neuronal circuits that are pathological? Well, one example are pain circuits. So neuropathic pain, for example, are pain circuitry that you can't quiet down, you can't suppress. And so one of the peptides from the nirvana cabal of Conus geographus has reached human clinical trials for intractable pain. And it turns out to be a neurotensin homologue, and the snail has evolved a neurotensin-like molecule, which is very unusual because it has a post-translational modification. It's glycosylated. Another nirvana cabal peptide that has reached human clinical trials is the sleeper peptide that, in fact, Craig Clark isolated many years ago. And this has been developed for intractable epilepsy. And it turns out to be a subtype selective NMDA receptor antagonist. So as you can see, if you know what the snails are using the peptides for, sometimes you can infer what a possible therapeutic application could be. So far, I've discussed peptides in a biological context, how they're used by the snails. And I'd like to shift gears a little bit and talk about why individual peptides can be very scientifically interesting. And to illustrate that, I'm going to talk about a family of ion channels called nicotinic acetylcholine receptors. So when you smoke, nicotine is, of course, a neuroactive substance, and many of the effects of nicotine are pleasant, and some are not so pleasant. And these are all mediated by a group of receptors called the nicotinic acetylcholine receptors, which are, in fact, ion channels that are activated both by nicotine and the neurotransmitter acetylcholine. So the complexity in this family arises from the fact that there are many genes. And so this shows you examples of those genes. You can split them into alpha subunits, alpha 1 to alpha 10, and non-alpha subunits, beta subunits, gamma delta. But the real complexity, potential complexity, is that the functional nicotinic receptor is a pentamer. It has 5 subunits. And those 5 subunits give that particular ion channel its specific properties. And so what you see are 5 examples of nicotinic receptors. And we know that the one shown here is, in fact, found at the neuromuscular junction. And so that's the nicotinic receptor that receives the signal from the motor axon. But shown are 4 different examples of nicotinic receptors that are found in the nervous system. And the problem is we still don't know how many different nicotinic receptors there really are. And one of the things that Conus peptides are helping us to understand is what are these different nicotinic receptors doing? So it turns out that a particular Conus peptide that binds a nicotinic receptor binds at the interface typically between an alpha and non-alpha subunit. And so here you see an example of different Conus peptides and their binding sites. And so you might have one that's specific for, let's say, the alpha-4 beta-2 interface or the alpha-6 beta-2 interface. And therefore, that's how these particular peptides get their specificity. Now, the reason why this is useful is because here are some nicotinic receptors, different combinations, and we really don't know where these are found and what they do. And so the way in which we try to figure out what a particular molecular component does these days is primarily genetic technology at a set of principles called gene knockouts. And so by knocking out a gene, you begin to figure out what that thing is doing in a physiological context. And here's the problem. If I wanted to figure out, let's say, what this particular nicotinic receptor did, then I might try to approach that by doing a gene knockout. So then I would knock out the beta-2 subunit, and that would get presumably rid of this complex. But what you can see is that if I knock out beta-2, I knock out all of these because they all have a beta-2 subunit. And so then if you get an animal, let's say a mouse or a zebrafish that lacks the beta-2 subunit, you typically have this very complex phenotype, and you really don't know which of these different ion channels was the ion channel that gives you that specific phenotype. And so you need to complement genetic technology with pharmacology, and that's where cone snail venoms come in, because they have a specificity in, which has the potential to differentiate between these different molecular forms. So let me show you how this works in a biomedical context. So it is known that smoking protects against Parkinson's disease, and the heavier you smoke, the more protected you are. But there are two problems. One, of course, is that smoking has bad effects, and that if you stop smoking, you lose all your protection against Parkinson's disease, but none of the bad effects of smoking. So it's not ideal to try to prevent Parkinson's disease by heavy smoking. It turns out that we want to understand why smoking is protective, because it's the nicotine in tobacco that seems to be protective. And long ago, my colleague Michael McIntosh, who was the undergraduate who discovered Prialt originally and is now a professor of psychiatry, he found this peptide, alpha-conotoxin M2, which when he labeled it, labeled an interesting region in the brain, the dopaminergic circuitry, precisely the circuitry that begins to become pathological in Parkinson's disease. And so what you see is an autoradiogram of the labeled peptide, and it corresponds to the same region that an antibody against the dopamine transporter labels. What Michael realized was that this peptide was not so specific for mammalian channels and hit the three channels that I'm showing here. And so what he did was he did structure function, and he was able to get derivatives of the peptide that were more specific. And so he was able to show pretty early that the alpha-3 beta-2, the top left ion channel, was not present in dopaminergic circuitry, but that in fact the two forms that have alpha-6 were present in the dopaminergic circuitry by using these particular Conus peptides. So you might say, well, who cares whether you have the nicotinic receptor on the left or the nicotinic receptor on the right? They're almost identical, right? They only differ because one of these has two alpha-6 subunits and the other one has an alpha-6, alpha-4. Well, you might care if you have Parkinson's disease. And the reason is that it turns out that although both of these are present in the dopaminergic circuitry, the one on the left does not decrease in level as Parkinson's disease develops, but the one on the right disappears very early on. We don't know yet which is the receptor that really helps because of smoking, but we do know that these two different nicotinic receptors behave very differently. And so that turns out to be interesting. We're hoping that we can develop perhaps a diagnostic for Parkinson's disease by looking at the nicotinic receptor that disappears early so that we can monitor people who are going to get the disease before any of the symptoms begin to kick in, which is often when 80% of the dopaminergic neurons have already disappeared. Now, here are two more nicotinic receptors. These are the so-called all-alpha nicotinic receptors, and we found peptides that can differentiate between them. Again, fairly small peptides. And what's interesting is these peptides don't come from fish hunting cone snails. They come from worm hunting cone snails. And so this group of worm hunting cone snails, their favorite prey are a type of worm that if you've ever been scuba diving, you're told, never touch this. These are called fireworms. And they have spicules that are extremely painful. And yet somehow these snails, that's their favorite meal. They're specialists on eating fireworms. And so in this group of snails, that's where we found a lot of peptides that are very specific for those particular nicotinic receptors that only have alpha subunits. And so we are able, therefore, to get a range of peptides that can differentiate between the two green nicotinic receptors shown here. And we think the reason why we find these in worm hunting cones is because in invertebrates, in contrast to vertebrates, it's these all green nicotinic receptors that you tend to find in the neuromuscular junction. And so these are paralytic to worms. And that we can then use to look at the same types of receptors in vertebrates. Well, what's interesting is something unexpected popped up once we had a peptide specific for the alpha-9, alpha-10 receptor, which is this receptor shown here. And it turns out it's anti-nociceptive. When you injure a nerve, you usually get a pain syndrome. And if you inject this peptide, you're able to prevent that pain syndrome. And so this actually has some biomedical potential. So as you know, people who get injured nerves, especially war veterans, very often have severe pain syndromes that are difficult to treat. And so this is one promising approach. But we didn't know that this particular receptor played this kind of physiological role. By having a peptide that's very, very specific, we now know that this nicotinic receptor plays a role when you have a nerve injury. And apparently what happens is when you have a nerve injury, you get an inflammation. And so there are immune cells that begin to attack the site of nerve injury. And it's that attack by the immune system that causes the pain syndrome. Now, what's interesting is that if you block that alpha-9, alpha-10 receptor, you prevent the immune system from attacking the nerve, the injured nerve. But if you block the closely related nicotinic receptor called alpha-7, because we have a peptide specific for that, you make the attack even worse. What happens is you get an even greater inflammation and more immune cells accumulating at the site of nerve injury. And that's why it's so important to have very, very specific pharmacological tools to differentiate between these different receptors. So I've shown you that cone snails are a biodiverse lineage, and really their evolutionary success is because they have a great delivery system and they have rapidly evolving gene superfamilies that allows them to develop pharmacological agents for each ecological niche. And so in each conopeptide superfamily, structure is conserved, but new function evolves extremely rapidly. That allows us then to take advantage of the very high selectivity and specificity of these peptides to study different molecular components in excitable cells. So I'd like to close by acknowledging the people who actually did the work and my collaborators, and the videos of the snails were taken by Jason Biggs. Thank you very much.